Hello everyone, welcome back to Intuitive Epidemiology. In today's video, we will unpack a research paper in epidemiology. My name is Taylor and I am an epidemiologist. Before we dive into the paper, I will recap some key concepts from previous videos. Epidemiology is the study of the distribution and determinants of health-related states or events in specified populations, and that is the application of the study to the control of health problems. In epidemiology, we focus on distributions or occurrences, on determinants, which we often refer to as exposures, on states or events, outcomes, and we examine these factors in specified populations or study samples. Epidemiology can be broken down into three broad analytical approaches: description, prediction, as well as explanation. In the context of explanation, we examine relationships or quantify associations between exposures and outcomes. And this can be most easily visualized using what is known as a 2x2 table. In this table, you can see how many exposed people did versus did not go on to experience the outcome, and how many unexposed people did versus did not go on to experience the outcome of interest. In terms of quantifying a relationship between two variables, in this case an exposure and an outcome, we can use measures of effect. In a previous video, we discussed the odds ratio, and in the paper I will present in this video, you will see risk ratios. A risk ratio is a probability of the outcome occurring in the exposed group divided by the probability of the outcome occurring in the unexposed group. An odds ratio or a risk ratio of 1 indicates that the exposure has no impact on the occurrence of the outcome. An odds ratio or a risk ratio greater than 1 indicates that the exposure increases the likelihood of the outcome occurring. And an odds ratio or a risk ratio less than 1 indicates that the exposure decreases the likelihood of the outcome occurring. A concept previously introduced, which is very important to the validity of epidemiological studies, is confounding. Confounding are differences in the exposed and unexposed group beyond the exposure itself. And a confounder or a confounding variable represents the factor which differs between the exposed and the unexposed group. In the case of an observational study, like the one I'm about to show you, there are typically many confounding variables that must be considered in a given analysis. As discussed in detail in past videos, observational analyses often involve multivariable models, also known as adjusted models, in which case we can use a statistical model to examine the relationship between our exposure of interest and a given outcome. And we can estimate our measure of effects from this model. For example, a measure of effect could be an odds ratio or a risk ratio. In the paper that I will show you, you will see risk ratios, and you can also get an associated measure of variance or a measure of precision known as a 95% confidence interval. We will discuss the concept of 95% CIs in a later video. By adjusting for confounders in a multivariable model, you can eliminate the effect of the bias that may be due to that confounding factor. Now, this is the paper that we will go through today. As you can see, I authored this paper and it was published back in 2018. This paper was completed during my PhD thesis at McGill University, and it is titled Injection Drug Use, Food Insecurity, and HIV/HCV Co-Infection, a longitudinal cohort analysis. First, I will highlight the structure of an epidemiological study, or the different headings that exist within a scientific publication in the area of epidemiology. First, we start off with an abstract, which is a summary of the paper. Then we have an introduction section where we justify the need for this study. while also highlighting past literature that is related to the study at hand. Then we have a methods section where we break down the study population, as well as the measures or the data that we are collecting from participants in order to complete our study. We have a data analysis section where we talk about how we've prepared our data for our analysis. We also talk about the specific statistical models, more specifically the regression models that we use in order to estimate our measures of effect. In this breakdown, I will gloss over these ideas, but indeed, if you want to read the full version of this paper, I've attached a link to it in the description in this video. We also have a results section, which is typically made up of text, as well as tables and/or figures. In this paper, there are several tables that outline the results in addition to a few paragraphs of text. This is the first table, which we will revisit in a moment, as well as the second table. We then go on to have a discussion section where we discuss our results in the context of the broader evidence base or in the context of the other literature that is based on a similar topic area. And of course, we have a strengths and a limitations section, as every epidemiological study has many strengths, but it also can have many weaknesses that have to be considered in order for a reader of the study to properly contextualize the results or the conclusions. And of course, we have an acknowledgement section where we typically thank our funders as well as collaborators on the work. Now, we will return to the abstract at the end of this video, but I'll first walk us through the introduction. So here we justify why this study is being done. We talk about food insecurity, or FI, as being highly prevalent among people living with HIV, hepatitis C virus co-infection. This is our study sample or our target population. We also talk about the exposure, injection drug use, or IDU, as being a primary risk factor for becoming co-infected with hepatitis C virus as well as HIV, as these are both blood-borne viruses. We also talk about food insecurity being associated itself with poor outcomes, such as suboptimal HIV treatment adherence, so poor treatment adherence, incomplete HIV viral load suppression, which is something we do not want in the context of HIV infection, and lower CD4 cell counts, which are lower immune cells in the body. So because of the high prevalence of the outcome, food insecurity, the exposure, injection drug use, and consequences of being food insecure, we analyze these data in a longitudinal fashion to examine the relationship between injection drug use as the exposure and food insecurity as the outcome among individuals living with HIV, HCV co-infection in Canada. So that's the justification for this work, which in this paper is quite brief. We then move on to the methods section where we talk about our study population. I'm going to skip through this section, but more details can be found here of how we had our study sample established and who we eventually collected data from in order to complete our epidemiologic analysis. Then we talk about our measurements. So we talk about capturing information on use of injection drugs in a 6-month window. And this is our exposure variable. You can also see here that we looked at injection drug use frequency, which is essentially just another way of coding the exposure variable. It's another form in which we can look at the exposure or the behavior of injection drug use. However, I won't focus on this second coded version in this discussion. We talk about food insecurity, our outcome, FI. And again, there's several ways you can code food insecurity or look at this outcome, but I won't get into those details here. But again, the paper is linked below if you'd like to look at this further. And I also highlight that we collected information on various confounding factors, or factors which may bias our examination of the relationship between injection drug use and food insecurity. The data analysis section can be quite technical, and that will not be the focus of this video. But I did want to emphasize that we describe the participants, and then we complete our statistical analyses, where again, the outcome here is food insecurity, and we're using a statistical model to estimate our measure of effect, quantifying the relationship between this exposure, injection drug use, with the outcome. In this case, we're estimating adjusted risk ratios. Again, we looked at various forms of the exposure in this paper, but that's not important to the takeaway messages in this video. In terms of the results, as I said, epidemiology is about the distribution or the occurrence of our exposures and our outcomes. Therefore, I discussed that 54% of participants experienced food insecurity at their first study visit, and that nearly half experienced severe food insecurity. I also mentioned that 31% of participants engaged in injection drug use, which in this case is the exposure. Now let's walk through Table 1, descriptive characteristics of the 608 co-infected individuals at their first time point in the study sample. We have the exposure variable, injection drug use, and we break this down by our outcome variable. This is known as stratifying our counts, our occurrence of the exposure by different levels or severities in this case of food insecurity. We also break down our socioeconomic, sociodemographic, behavioral, and clinical confounding variables in this Table 1. This gives us a better sense of the characteristics of the study sample, which prepares us to look at Table 2. Table 2 highlights the regression models or the statistical models that we've used to quantify our relationships between the exposure and the outcome. We quantify that relationship using risk ratios as our measure of effect, and we highlight that the exposure is injection drug use and the outcome is food insecurity among the 608 co-infected participants between 2012 and 2015. This is a longitudinal analysis, meaning that we actually have measurements on substance use and food insecurity on participants repeatedly over time. Again, we look at the exposure in various forms, different numbers of categories or ways to break it down. The takeaway message is that injection drug use, after adjusting for all of the aforementioned confounders, which you can see here, is associated with the outcome because the risk ratio after adjustment is 1.15. The number you see here is known as a 95% confidence interval, which is closely tied to statistical significance, and we will unpack this concept in a later video. The takeaway is that the exposure's coefficient, or the measure of effect in the model for injection drug use on the outcome of food insecurity, is 1.15, which is greater than 1, indicating that engaging in injection drug use is associated with a higher likelihood of experiencing subsequent food insecurity. These results are reiterated in the text, IDU, injection drug use, being associated with food insecurity. And then we go on to discuss these results in the broader context of other literature and other papers that have looked at this topic within other study samples. We start off by indicating that we found evidence of an association between these two factors. Again, this Table 3 is just another way of looking at food insecurity, where we look at a more granular categorization. We will not focus on this table in this video. And of course, strengths and limitations. Because we are completing an observational analysis, As discussed in the past video, this is not a randomized controlled trial, in which case we have not randomized the exposure, as that would clearly be highly unethical. We have to discuss limitations that are inherent to observational study designs, such as cohort studies, which is what this study is. One of the main limitations is unmeasured or imperfectly measured confounding variables. So while I adjusted for a variety of socioeconomic, sociodemographic, behavioral, and clinical factors in order to eliminate confounding bias by these factors, this is an imperfect strategy, as in the absence of randomization, you cannot ensure that you've adjusted for every single confounding variable, which means that confounding bias may remain, and our risk ratio, that number 1.15, while we think it is valid, may still be biased by confounding. We conclude that the association between these two factors appears to exist in our data, and that independent of socioeconomic, sociodemographic, behavioral, and clinical confounders, there appears to be a link between injection drug use and food insecurity among people living with HIV, hepatitis C virus co-infection. We'll go back up to the abstract, the summary of the paper, and you will see all of the same ideas summarized here. And with that, I would like to thank you for watching this video. If you found it useful, please subscribe to this channel. If you have any questions or comments, feel free to leave those below, and please check out related content on this channel.